Okay. So the basics of molecular biology, right? Protein, RNA, or DNA to RNA. We talked about replication, this conservation of this material and the inheritance of this material by other cells. We're now going to talk about this transcription process. Transcription is DNA dependent, RNA synthesis. Reverse transcription, RNA dependent, DNA for synthesis. We talked about that. So how does transcription begin? How is transcription initiated? These figures are from your text. It's generated from E. coli. In E. coli, there's an enzyme that's called the whole enzyme because the whole enzyme has a catalytic subunit in brown as well as a subunit. It's called sigma. It's responsible for identifying the promoter. This picture shows that complex sitting down on a promoter. That's not true. It actually sits down on the DNA. And when it sits down on the DNA, I label it step 0 because they give that step 1. But when it sits on that DNA, it has a relatively low affinity. So the sigma factor has a relatively low affinity for all DNA. And its affinity, it has a KD of about 10 to the minus 4 molar. So which once that complex sits on that DNA, it's able to scan or to walk or to move up the DNA. And it moves along that DNA until it reaches a promoter. And when the sigma factor reaches a promoter and binds to that promoter, the KD changes tremendously to 10 to the minus 9. So now this guy has a high affinity for the promoter region, and he stops. So the idea is that polymerase gets on the DNA, moves along until it finds an open promoter, and it stops. Once it does, the next thing that happens is it opens. This is called a closed complex, by the way. Once it does, it begins to melt that DNA. That DNA is melted for about 15 base pairs. It's called an open complex. The footprint actually is a little different because this, even though this looks like it's covering where that leaves is for 15 pairs, maybe that's supposed to be open more. But the footprint is about 90 nucleotides, so it's a pretty big complex that sits on that DNA molecule. The unwinding process doesn't require energy. As soon as that sits there, it seems to begin to unwind it. Once the open complex forms and there's this open loop, RNA synthesis begins. RNA synthesis begins. It's called this original process is called initiation. It's the initiation step. And when the initiation step goes on, this guy begins to make nucleotides. And the process, I don't know that your book tells you very much, but the initiation process actually is somewhat hit or miss. The reason is what happens is as this newly synthesized, or this nascent RNA chain is being synthesized, it forms a complex with the DNA. So there's actually a hybrid in here between the template strand, doesn't show real well, but the template strand and the new RNA molecule. And that complex or duplex is about averages in E. coli, about 12 nucleotides long. It's between 10 or 15 or so nucleotides long. And remember, when you form a complex between two oligonucleotides, at least RNA and DNA, you need about 7 of them to have any stability at all for that binding. So when RNA synthesis begins and it starts to make an oligonucleotide chain, some of them actually fall off because the RNA is not hybridized or it's not duplexed with the DNA. So that process is kind of it just keeps going until finally it makes 10 and it forms that duplex. But it's very important that about a dozen or so nucleotides form this. Once that happens, or as that's happening, as that initiation process is happening, this whole complex then stops. It just stops dead. Why do you think it stops dead after about 10, 12 nucleotides or so? Why do you think it stops? So that in order to find this complex, or this promoter with the sigma factor, remember we've gone from fairly low affinity to extremely high affinity. In order to have any specificity to identify promoters, there has to be quite a high affinity between that recognition factor, sigma, and the promoter. So once transcription begins like this, and you start getting about 10 nucleotides or so, everything kind of comes to a stop because the complex, this transcription complex, cannot pull away from the promoter region. It's just bound very tightly. Okay? So, and it has to let loose. And the way it does is that this holoenzyme that has the catalytic sites as well as the sigma factor, right, actually jettisons the sigma factor. So the sigma factor comes off, and we now have this core enzyme. You can see the core enzyme is just a catalytic site. There's really not any more affinity for the promoter. So now, once we've initiated transcription, in the initiation stage, we can enter elongation as soon as sigma's gone. And so the complex is able to move up the DNA and copy that DNA. This is just a picture that shows you this. We've looked at that before. It's sort of interesting. Remember in DNA polymerase, when they did the crystal structure, whoever did it called this thing the thumb. Here they call it the letter. It's the very same thing. Both of the crystal structures of both the RNA and DNA polymerases have this structure in this is this group right here, the rudder, which actually fits in between the two strands of DNA and helps pull them apart. So as the DNA moves through, it actually pulls it apart. Here you can see the blue is the new strand, and the new strand sort of fits under this region here, right? It's sort of flat, it closes on it, so it sort of helps to hold it in. But what really holds that RNA in is right in here, where the template is duplexed for about 12 to 15 nucleotides or so with that RNA. And interesting, as long as that stays duplex like that, transcription continues. And both the eukaryotes and prokaryotes, the termination of transcription essentially is the dissolution of that duplex. If you remove the duplex, RNA molecule falls off and transcription ends. We'll talk about that in eukaryotes at least in a minute. I think that's all I really want to show you. You can see the picture with the double stranding coming in here, unwinding here, and then it has to rewind on the other side. So you need a helicase on both ends and a gyrase on both sides to unwind and rewind that DNA after it transcribes. What's in the promoter that makes it stop? You've probably seen this picture many times. This is a picture of the promoter region in E. coli, but it's similar to many bacterial cells. Promoter region is the region that's upstream, the 5' side of the gene. So here's an initiation site. This is the beginning of the transcription right here. And upstream seems to be numbered negatively, right? So this is minus 154, there's minus 35. But upstream, if you line up many genes, what you're seeing is there are regions that essentially coincide with each other. The one that's called the Pribnow box, or the Tata box. The center of it is about minus 10, so it's about 10 nucleotides from here to the start site. That Pribnow box, TAT, AAT. The upstream from that is the minus 35 region. There's another box, which is TTG-ACAT, TTG-ACAT. This region up here is also conserved. If you look at it, it's conserved. Now, there's a couple of things. First of all, why do we call these consensus sequences? They're called a consensus sequence. Well, it is because if you look at this, at these regions up in here, many genes, although you can see attached to the Tata box, they're not perfectly conserved, right? There's a CAT, there's a TAG, there's a CT, there's a GT, so they're differently conserved. But if you look at take an average of all of these, right, this is the consensus, TAT, AAT. It's 9% of all, from all of the promoter regions, it's going to be a T, 90%, 5%, that's an A, 96%, the last one is a T. You can see pretty much a T all the way there. So the consensus sequence is the sequence that average all of this, all of these. Same thing here, here's a consensus sequence, numbered not quite as strong. The TTG are the strongest in there, but then they're the most conserved. What is the function of this consensus sequence of this promoter? What's the function of the promoter, first of all? What's going to happen there? We just talked about what happens at the promoter. The replication complex, right? The transcription complex, not the replication, but the transcription complex.